Good day, everyone. I am Gicel Paclibar from the Lyceum of the Philippines University. Today, I will be discussing our paper entitled Allelopathic Effects of Lantana camara Aqueous Leaf Extract on the Seedling Germination of Zea maize. Invasive alien species is one of the major threats of biodiversity. Lantana camara is one of the most widespread invasive weed species, which is rampant here in the Philippines and were already established in disturbed areas and many agricultural areas. Lantana camara is used as an ornamental plant, and it also serves some medicinal purposes as it contains antibacterial and larvicidal properties. Studies about the allelopathic effect of Lantana camara that had inhibitory effects were recorded in agricultural crops, bryophytes, forest crops and communities, and even to other weeds. In line with this, stimulatory effects of Lantana camara were also recorded in some agricultural crops. Allelopathic effect of Lantana camara in maize were studied by Tadele in 2014 and Yu and Raha in 2015, Mawal and Patil in 2019, and Gautam et al. In 2021. However, no accounts were available in using the full concentration of the Lantana camara leaf extract. Zea maize or corn is one of the most important food source and agricultural crop in the Philippines. Zea maize is recognized as the second principal agricultural crop next to rice and plays an important role as one of the major sources of livelihood for Filipino farmers. Since weed infestations are recognized as a serious biological constraint in crop plantations, further studies about the allelopathic effect to agricultural crops must be conducted. The main objective of this study is to evaluate the allelopathic effect of Lantana camara in the seedling germination and root and shoot elongation of Zea maize. Specifically, the study aims to determine the significant differences of the effect of Lantana camara aqueous leaf extract concentrations to the seedling germination and root and shoot elongation of Zea maize. Next, identify the concentration with the most inhibitory activity. And lastly, determine the effect of the presence and absence of aqueous leaf extracts to the Zea maize. For the extract preparation, mature leaves were collected in Biluso, Silang, Cavite, where it already proliferated in the wide farmland. The collected leaves were disinfected using ethanol and were air-dried for 15 minutes. The leaves were then processed using the preparation performed by Tadele through immersion of 100 grams of leaves of Lantana camara into 1,000 ml containers filled with 500 ml distilled water, which are then sealed in an Erlenmeyer flask and later shaken and stored at room temperature for at least 24 hours. After storing, the aqueous leaf extract were filtered using filter paper and diluted into 25, 50, 75, and 100% concentrations. The extracts were stored in Erlenmeyer flasks, sealed with a farafilm, and kept in the fridge. For the germination and growth records, the seeds of maize were washed with distilled water and surface sterilized with sodium hypochlorite aqueous solution for at least 5 to 10 minutes to avoid contamination and had been rinsed thoroughly with distilled water multiple times while the control had been treated only with distilled water. 15 sodium hypochlorite treated seeds of maize were evenly placed in 9 cm diameter Petri dishes lined by Wattman No. 1 filter paper with 3 replicates per treatment. The seeds were then treated with 5 ml of 25, 50, 75, and 100% aqueous extract concentration and 5 ml of distilled water for the control, which were applied daily with one dosage applied from 8 to 9 a.m. in the morning and a second dosage at around 3 to 4 p.m. in the afternoon to keep the moisture within the filter paper. This had allowed seedling development for 14 days period. The prerequisite for considering a germinated seed is upon the appearance of the radicle. Germinated seeds were counted and checked daily, while the root and shoot lengths were measured at the conclusion of the experiment. For the data analysis, germination capacity and the root and shoot relative elongation ratio were calculated using the formula. For the germination capacity, GC in percentage equals number of germinated seeds divided by the total number of seeds sown multiplied to 100. And for the RER, or the relative elongation ratio of shoot, equals mean shoot length of tested plant divided by the mean shoot length of controlled, or the control, multiplied to 100. And for the RER of root, mean root length of tested plant divided by mean root length of control times 100. And the significant differences among the treatments were evaluated using analysis of variance, or ANOVA 1, and further assessed using the post-hoc 2-piece HSD test. The significant differences between the presence and absence of the aqueous leaf extracts of Lantana were calculated using t-test. So these are the seedling germination of Zea maize after 14 days. So for the seed germination, we have here figure 1 and table 1. For figure 1, the germination capacity of Zea maize in response to Lantana camara aqueous leaf extracts at different concentration. As you will notice, the germination capacity of the seeds of Zea maize was found to be highest at the control, followed by 25%, 100%, and lowest at 50 and 75% concentrations, with both germination capacity of 26.67%. For table 1, germination of Zea maize seeds in response to Lantana camara aqueous leaf extracts at different concentrations after 2 weeks, the number of seeds grown per treatment were also significantly different, wherein the control has the highest number of seeds grown and lowest at 50% and 70% concentrations. It is noteworthy that 25% concentration of Lantana leaf extract has no significant difference with a control, and 50%, 75%, and 100% concentrations had no significant differences, indicating that high concentrations of Lantana aqueous leaf extracts negatively affect the seed germination and the germination capacity of Zea maize. In line with this, 75% concentration of Lantana camara leaf extracts had the most inhibitory effect to the germination of Zea maize. Same results were observed in agricultural crops such as spinach and cucumber seeds, where full strength of Lantana camara extract did not significantly inhibit its germination. For the root and shoot elongation of Zea maize, we have here two figures. For figure 2, the relative elongation ratio of Zea maize roots in response to different extra concentrations of Lantana camara after 2 weeks. And on the other side, the figure 3 is the relative elongation ratio of Zea maize shoots in response to different extra concentrations of Lantana camara after 2 weeks. The relative elongation ratio of root reveals that Zea maize roots that were exposed to different aqueous concentrations of Lantana camara had relatively lower RER than the control, having the 75% concentration as the lowest, followed by 100%, 50%, and 25% concentration as the highest. And for figure 3, the result shows that the shoot of Zea maize that were exposed to the concentrations have lower RER compared to the control, in which the 50% had the highest RER, followed by 25%, 100%, and 75% had the lowest RER. Table 2 presents the root and shoot elongation of Zea maize in response to Lantana camara aqueous leaf extracts in different concentrations after 2 weeks. The longest length of root was observed in the control, while the shortest length of the root was observed in 75% concentration. And based on the findings, the control was significantly different among the treatments except in 25% concentration in terms of root elongation, wherein the higher the concentration is, the inhibitory effect of Lantana camara aqueous leaf extracts will also higher, making the root shorter in length. For the shoot elongation of Zea maize, the longest length of shoot was observed in the control and the shortest was observed in the 75% concentration. This indicates that Lantana camara concentrations inhibit the elongation of shoots of Zea maize. Based on the results, the control had significant difference among the treatments used, indicating that aqueous extracts of Lantana camara can inhibit shoot elongation. Lantana camara leaf extracts had negative significant effect to the elongation of both root and shoot of Zea maize, wherein the higher concentrations had higher inhibitory effects. The proportionality of the concentration and the inhibitory effects were in line with the study of Ahmed et al. In 2007, Hossein and Alam in 2010, and Carr et al. In 2014. It is also worth note-taking that the control differed to the other treatments except to 25% concentration in root elongation. Also, 75% concentration had the lowest length for both root and shoot with RER of 43.5% and 39.6% respectively, as I shown earlier. For the presence of Lantana camara leaf extract, Zea maize germination and root and shoot elongation were significantly different between the presence and absence of Lantana camara leaf extracts, indicating that Lantana camara leaf extracts had inhibitory effects on the growth of Zea maize, as revealed in the table that you can see. Having the absence of Lantana camara leaf extracts had higher mean as compared to the presence of Lantana camara leaf extracts. This result coincides with the study of Gicel and Duggan, wherein the removal of Lantana camara significantly increased the germination of 2 forest crops species. To add, leaf extract of Lantana camara was found to have a maximum inhibitory effect compared to the stem and root extracts. For the summary of my discussion, Lantana camara has economic uses but deemed to be detrimental to several agricultural crops because of its allelopathic properties. The study revealed that the different concentrations of Lantana camara aqueous leaf extracts had inhibitory effects in the germination and root and shoot elongation of Zea maize, wherein the higher the concentration is, the higher the inhibitory effect the concentration will cause to the Zea maize growth. Additionally, the presence of the Lantana camara leaf extracts had significant difference to its absence on Zea maize growth. Because of this, further studies must be conducted to test the allelopathic effect of Lantana camara to other agricultural crops here in the Philippines. Thank you for paying attention to my discussion about our research study. Thank you again and have a pleasant day, everyone.